Hi there. Today we'll be learning about glycolysis and its intermediate three steps. So let's first go through what glycolysis actually is. In simple terms, it is the first step of respiration in any living organism and it occurs in the cytoplasm. So when you think about respiration, you would think that it occurs in the mitochondria as it breaks down oxygen, but the first step takes place in the cytoplasm. So let's say this is the cell. As the glucose molecule enters the cytoplasm of the cell, it undergoes glycolysis. So the word glycolysis literally means the splitting, so lysis means splitting of glucose. So glycolysis, so the splitting of glucose. And now we're going to see what the splitting actually means. So let's get a big picture over here first. We input two molecules of ATP, and from there, as the pathway runs, we get a total of four ATP molecules. So that's a net gain of two ATP per cycle, per pathway. And along with that, we also get two NADH molecules. So NADH is again another energy rich compound. So let's begin with glucose. So glucose undergoes a change by adding a phosphate group. So we go from glucose to glucose 6 phosphate, where the phosphate group is added on the sixth carbon under the enzyme hexakinase. And here we use one ATP molecule. So we take one phosphate group from the adenosine triphosphate and convert it to adenosine diphosphate. Now that we have a glucose 6 phosphate molecule, it undergoes a isomerase change. So there is no addition, there is no subtraction, there is just a change in the structure of the molecule. So that is catalyzed by phosphoglycerate isomerase, and as you can see, it has the word isomerase in it. So it clearly tells us that there is only a physical change occurring. So now we have a fructose 6 phosphate. Now, just a reminder, everything that we've seen till now, from glucose to fructose 6 phosphate, it's all a 6 carbon molecule. So glucose is C6H12O6 and so on, and all of these contain 6 carbon atoms. Now, in our third step of the pathway, we go from fructose 6 phosphate to fructose 1,6 biphosphate. So, again, we have another addition of a phosphate group, this time at the first carbon in the fructose molecule. And again, we require one more phosphate group, which we take from the adenosine triphosphate in the cytoplasm. Now, this is catalyzed by the phosphofructokinase 1 enzyme, and after this is where the major split happens. This is where we go from a 6 carbon molecule to a 3 carbon molecule. Well, actually, two 3 carbon molecules for every glucose molecule. So let's go through dihydroxy acetone phosphate. And so DHAP is converted, fructose 1,6 biphosphate under aldolase enzyme is converted into two products. So the first is a 3 carbon molecule, DHAP, and the other is also a 3 carbon molecule, which is glyceraldehyde 3 phosphate, so G3P. And DHAP is converted to G3P by triose phosphate isomerase. So there is no addition, there is no subtraction. Again, it is just a isomerism change. So now at this point, after all this is done, we have exactly two molecules of G3P. And now we're going to remove a hydrogen group from here and get under the enzyme glyceraldehyde hydrogenase. So now, as you can see, we have two of these because the first is converted by aldolase directly, and the other is through DHAP and then trisphosphate. So now this molecule is a 3 carbon molecule. So 1,3 biphosphoglycerate is a 3 carbon molecule with two phosphate groups, one at the first carbon and one at the third. And right before this, we are taking two NAD plus molecules and two molecules of inorganic phosphate, and we convert them into two molecules of NADH. Now we go through another change. Here, before, as you would see that in these steps, we were adding a phosphate group from adenosine triphosphate to make it diphosphate. So glucose would gain a phosphate group and become glucose 6 phosphate, and fructose would gain fructose 6 phosphate would gain a phosphate group and become fructose 1,6 biphosphate. But here, on the other hand, we are taking away a phosphate group and putting it back into the cytoplasm. And when that happens, since we are having two molecules of 1,3 biphosphoglycerate, we can give it back. We can give two molecules of phosphate to back to the cytoplasm, which converts two molecules of ADP into two molecules of ATP. And at this step already, we have filled or we have returned the exact amount of ATP back to the cytoplasm as we've already used. So one over here and one over here. And now any ATP that we produce is going to be a net gain. So let's see how that goes. Now we go through another mutase change. So we go from 3 phosphoglycerate to 2 phosphoglycerate. So now the phosphate group is at the second carbon. Remember, from the fifth step onwards, all our products are twice because these are all 3 carbon molecules as compared to the 6 carbon molecules that we began with. Now there is a change from 2-phosphoglycerate to phosphoenol pyruvate or PEP under the enzyme enolase, and that is further converted to in a final step into pyruvate, which is our main product and the main catalyst that we use to enter the Krebs cycle. And as a PEP is being converted to pyruvate by pyruvate kinase, we gain back two molecules of phosphate back into the cytoplasm. And what that does is that it creates two molecules of adenosine triphosphate. So by the end of the glycolysis pathway, we have created, let's write this down here, we have created a net gain of two ATP molecules and a net gain of two NADH molecules, along with two molecules of pyruvate, which is a 3 carbon molecule. So, for every glucose molecule that enters the cytoplasm, we get the following results.